Now let's start our study of DNA fingerprinting. The term itself suggests that what does it define? Fingerprinting that is carried out using a DNA sample or with the help of DNA. The fingerprint that is carried out is known as DNA fingerprinting. This process or DNA fingerprinting technique came into existence and is used by detectives and police labs during the early times, that is 1930s, to understand the specific nature or speciation of a person that had been involved in a crime. Thus, in this manner, the crime suspect can be arrested with the help of this technique. This figure depicts the types of fingerprint. This figure is the DNA fingerprint and this one conventional. Conventional means the fingerprint that was carried out during the early time. Unlike conventional, the DNA fingerprint can be carried out from the DNA cells that is present within every cell, tissue or organ of a person. But on the contrary, in conventional fingerprint, the fingerprint can be carried out only with the help of the cells that are present at the fingertips. Consequently, DNA fingerprinting became rapidly famous for identifying and distinguishing the individual human beings. In this manner, they can be individually and distinctly identified on the basis of fingerprints of modern technology, that is DNA fingerprinting technology. Moreover, it also helped in diagnosing various hereditary diseases. Now, as we all know that in humans, there are mainly 23 pairs of human chromosomes with about genome size greater than 3 billion. However, all the DNA segments are regulatory in function. This we have already studied during the regulation of gene expression. While there are a few sequences which occurs in the form of introns or occurs as a repeated DNA sequence. However, for DNA fingerprinting, there are specialized nucleotide sequences known as short, repeated, nucleotide sequences which are called as variable number tandem repeats. Whose short form is VNTR. Thus, variable number tandem repeats can be defined as short repetitive nucleotide sequences which are specific for a person and helps in carrying out the DNA fingerprint. This concept of VNTR was first developed by Eno Jeffries, where he used a satellite DNA as probe that showed a very high degree of polymorphism. That is, within the organisms of the same species, different phenotypic structure occurred due to this DNA probe that was present within the chromosomes. On this basis, the concept of DNA fingerprinting using variable number tandem repeats was found. Thus, in this manner we can say that DNA fingerprint is a quick way to compare the DNA sequences of any two living organisms. Apart from this, while carrying out DNA fingerprinting, it was found that the information that was contained in the DNA sequence was similar like that of letters in a zipper. This figure depicts the comparison of the DNA sequences within a gene to the segment of a zipper. This was found on the basis of DNA fingerprinting while studying the genome sequences within it. And thus, in this manner, it was easy to understand the DNA fingerprinting procedure in different living organisms.